This is an Exam 1 review for Bio 182. It was created by Dr. Tucker's undergraduate teaching team, composed of Francie, Justin, Katherine, Katie, and Mina. First, we're going to cover some genetics concepts, and then we're going to talk about some genetic sample questions. And then we'll cover some evolution concepts and go over some evolution sample questions. The links to the PowerPoints and sample questions are in the YouTube description. Hello, I'm Justin. We're going to be covering genetics. There is an accompanying PowerPoint that will cover the same information that we're going to talk about here, but because the exam is open note, it'll be a lot easier to reference information in the PowerPoint. We're going to start from the beginning with DNA, deoxynucleic acid. All living things have DNA, and DNA is composed of very, very long stretches of molecules called nucleotides that are linked together. There are 4 kinds of nucleotides. There's adenine, thymine, cytosine, and guanine. So these 4, when linked up in very long stretches, are able to produce a ton of different combinations. So DNA is able to store tons of genetic info for organisms or organism to read. So what do I mean by read? So the DNA is read as a blueprint to produce all the little different things that an organism needs within itself. A genome is your complete set of DNA. Cool. So let's talk about a gene, which is a section of genome which is composed of DNA that encodes a particular trait. So not all of the genome is particularly useful. In fact, for most organisms, a lot of the genome, the DNA, does not really produce anything. But there are some sections of the genome that can be read to produce a protein, or to ultimately produce a protein. So let's say, okay, so we're looking at a gene, right? Section of genome, right? Your genome is made of DNA. Your DNA is made of nucleotides. So maybe you have like A, T, 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 A, C, G, A, C, C, G, A, T, A. Okay? So let's say that this is a particular gene. It's extremely short, but what will happen is that it is read to produce mRNA. If you remember from last semester, this is called transcription. That mRNA is translated to ultimately produce protein. So this gene over here was read to produce the protein. And then, but we talked about traits over here. But this protein will ultimately produce the trait. So let's say that we're looking at the gene for hair color. So the hair color gene. And for this given a person's gene, it is read to produce the mRNA, and that mRNA is used to produce a protein that produces the hair color. Let's say that this is the blonde, this is for blonde hair color. So then that protein will ultimately produce the blonde trait. Blonde hair trait. Cool. So, yeah. So all humans have the same genes, more or less, but they don't have the same kinds of genes. And that kind of makes sense, you know, because if we're looking like, let's say that hair color was monogenic, meaning that there's only one gene that will determine it. You know, not all people are blonde, not all people are brunettes, you know. People have, they still have hair color, you know, so they still have the gene for hair color, but they don't have the same kind of gene. So these different varieties or forms of genes are called alleles. So form or variety of gene. So let's look at this one. Let's say that this is that was blonde, right? So now we have A, TAC, right? That once this is transcribed and translated, it'll produce the protein for brunette hair. I scribbled it because I don't know how to spell brunette. Anyways, yeah. So the gene was for hair color. That's the kind of trait, but there are different alleles for that gene. So this is kind of confusing some of the times. So I'm going to talk about an example. So let's think about cars, and let's kind of compare them to humans. So cars need engines. All cars have engines, I think. And then they have transmissions. And they also have wheels. Wow. So all cars have these traits. And so humans, they all have eye color. I mean, unless you don't have eyes. Whatever, let's not think about it too much. They all have hair color. And they all have, let me think. They all have fingers. Wow. Cool. So, but not all cars have the same engine. You know, that's what makes cars different. You know, that's what makes humans different. That they're different. But so one car might have a I don't know, I said freaking, not the other one, V8, or in another one might have I don't know, Honda Gen. Yeah, so those are different engines, right? And then, you know, they have different kind of wheels. You could have I don't know, snow wheels, and then you could have brown wheels. You know, cool. And then for humans, you know, you could have blue eyes, you could have hazel eyes, and then for fingers, you could have, you know, 5 on each hand, or you could be polydactyl, and you might have like 7. You know, so you can think of the different components of the car as the genes, and these are genes, right? So genes. And then you can think about the different kinds of these parts as the different alleles. Cool. Alright, let's talk about the difference between the genotype and the phenotype. So simply put, the genotype is your DNA, or your genes in particular, where your phenotype are observable traits. Now, these traits don't have to be seen by the naked eye, you know, like, I mean, hair color, height, those are observable traits, but it might be something like a protein within you. So, yeah, the phenotype and the genotype are never the same. And this is because, or I mean, across like everything, and that's because the phenotype is determined by gene interactions and also interactions with the environment. So, what do I mean by them never being the same? That means that even though you might have genes that are supposed to code for traits, you might not actually see these traits represented in the phenotype. So, there are 2 things involved in the phenotype. There are gene interactions. So just a sneak peek for the future. Like, think about like big A, little a. Even though you have that gene for little a, only the effects of big A will be seen because it's dominant. And let's look at interactions with the environment. So a great example of this are monozygotic twin studies. So these are identical twins who are separated at birth. The researchers love them. So let's say that they're like one twin went to a very wealthy household and the other one went to a household experienced poverty. So generally, they found that the twin who went to the wealthier household, you know, has better nutrition, probably better schooling, and that twin will have a higher intellect, will also be taller, will probably be healthier and more fit. So that is an interaction with the environment. So that environmental interaction resulted in different genes functioning and producing the trait. So another example would be type 2 diabetes. Even though one possesses the gene that produces type 2 diabetes, that gene might not be activated. So there are a couple of risk factors involved. So if one has a central mass distribution, has a sedentary lifestyle and maybe is a smoker, they have a much higher chance for that gene activating later on, being expressed, and producing the phenotype of type 2 diabetes. But a lot of people are genetically susceptible, if they live a healthy lifestyle, they won't experience that phenotype of type 2 diabetes. Your genome, the human genome, is not stored within one huge long strand of DNA. It's stored in multiple huge strong long strands of DNA. But it's not just in like a straight line, that would be crazy. It's compacted and is organized. And is organized into structures called chromosomes. So a given chromosome is made out of one strand of DNA and a bunch of different proteins. So these proteins help facilitate the organization and the folding of the DNA into a compact structure. Now humans have 46 of them. Other organisms might just have one chromosome. They just have their entire genome is stored on a single strand of DNA. Whereas other organisms might have more than 46 or less than 46. It really depends. Cool. So let's talk about chromosomes some more. Usually you'll see them depicted like this. And they're about the same length, and they're not trying to trick you, so they're just saying that these are the same chromosome. Let's say that we're looking at chromosome 1, and that this was taken from Juan, and this was taken from Mona. So this is essentially one huge long strand of DNA. And, remember how humans have the same genes? They just have different alleles? Well, the same thing applies with chromosomes. They have the same chromosome as well, but they just don't have the same nucleotide sequences all throughout. So let's start from the beginning, right? So after you look at a bunch of different nucleotides over here, eventually after a certain amount, you'll reach a stretch of DNA that codes for a certain gene. Let's say that for Juan, this is hair color, right? And he has little q, which would be blonde. And then for Mona, we go over here. After a certain stretch of DNA, we'll reach a stretch or a section of DNA that codes for hair color. And let's say that is for black hair color. Well, looking over here, looks like they're about equidistant. And that makes sense. So they're the same chromosome, so they have the same genes at the same locations. So you can start from the beginning, right? Let's say that you can call this one nucleotide 1, nucleotide 2, and eventually you reach nucleotide like 4 million, and then starting from that, you have the start of the hair color gene. Same for Mona, right? Eventually you reach nucleotide 4 million from the start, and that is where the gene for hair color is located, right? So it looks like they have different alleles here. So they have different nucleotide sequences. So Juan has the one for blonde hair, Mona has the one for black hair. Let's say we keep on going. Eventually we reach the gene for eye color. And let's say that Juan has black eyes, and Mona has black eyes too. So this is the gene for eye color, is at the same location, which makes sense because it's the same chromosome, and they just happen to have the same allele there. And now this is not, there are genes spread out all throughout on these chromosomes, and it's impossible for two organisms, unless they're twins, for them to have the same alleles at every single location. They might, so they have the same genes at the same location, but they have different alleles scattered throughout. But they're drawn at the same size because they are about the same size. Okay, let's talk about an example. So let's compare a human to an artist, a musician. So, for our human, let's call him, actually, I feel like girl names are a lot easier to come up with, So we'll go with, I've always liked the name Juniper. And then for the artist, I like the band System of a Down. Okay, so let's look at a human's genome, or Juniper's genome. And let's compare that to an artist's discography. So the genome is a complete set of all Juniper's genes, and for a System of a Down, it would be their complete discography, like all the music that they have produced. So let's say that you're looking for, so genome is made up of genes. Discography is made up of songs. And let's say that you're looking for a single CD that has every single one of their songs. Now, for official releases, that would not be possible because the songs are split up on different CDs, or I'm sorry, different albums. So there are 5 albums. You have you have their self-titled System of a Down. You have Toxicity. You have Steel's Album. You have my personal favorite, Mesmerize, and then you have Hypnotize. So it's spread across 5 albums. Cool. And in the genes, you could not find a single chromosome, like a single strand of DNA that has every single gene. Those genes are spread out across 46 different chromosomes. Cool. So remember that the chromosomes are not connected to each other. There are 46 strands of DNA that are compacted into the structure of chromosomes with the help of proteins. And yeah, so we have the same chromosomes as other people, more or less, and they can differ. There are 2 kinds of chromosomes that you need to know about: homologous chromosomes and sister chromatids. We're going to start with homologous chromosomes first. So, we talked about this in the video, in the thing. It's 4 o'clock in the morning. So, these 2 over here, whenever they show you things that look to be around the same length, that means that they are the same kind of chromosome. And if they're different colors, that just means that they have different alleles. So, same genes, different alleles on the chromosome. So, homologous chromosomes are just the same type of chromosome. So, this is a pair of homologous chromosomes. And this is another pair of homologous chromosomes. Now, let's look at sister chromatids. So, at the moment, we don't actually have any sister chromatids present. So, we gotta think about mitosis and meiosis when we're thinking about this. So, your body, you're making new cells all the time, and each one of your cells has a complete set of the genome. And what happens is that one cell during mitosis will split into an identical cell. Now, it can't just do this willy-nilly. It needs to first duplicate everything it has, and then it splits in half. And that ensures that both of the new cells created have a complete set of everything. And this includes DNA. If you didn't duplicate the DNA beforehand, you would just split it in half, and then as you would continue doing that, eventually you'd have a bunch of little cells, and none of them would have a functioning genome. They would have barely any DNA at all. So what happens? You gotta duplicate them. And so let's say that we're looking at this chromosome over here. So we need to duplicate this one. What'll happen is that as you duplicate a new chromosome, the chromosome will immediately stick to the old one. This is the new one, this is the old one. So this is called a sister chromatid. So it's still 2 chromosomes, but they're identical to one another. So that's why the colors become kind of useful. So over here we can see that they're about the same length, so they're the same type of chromosome, but not only that, but they're the same color. That means that they have the same everything. They have the same nucleotide sequences. That means they have the same alleles. Like over here, so this one is homologous to this one. This one is also homologous to this one, but these these 2 that are connected to one another that we call a sister chromatid is literally identical. So the sister chromatids are same exact chromosome. So usually you'll find them in this X-shaped form. And then after they're separated, so then this will go into one cell, and this will go into another cell. Yeah, so usually sister chromatids, you'll find them in that form, they're made for that purpose of replication, and then they're separated. Ploidy is the number of sets of chromosomes that an organism has. So we are diploid. Di. 2. So we have 2 sets of chromosomes. We have 46 chromosomes present, and then it's present in 2 sets. So 23 chromosomes and then 23 chromosomes. That means that, okay, we'll talk about this in a little bit, but let's say that we're looking at the genome of a human female. So, there are 23 homologous chromosomes. So, what does that mean? That means that, okay, so let's say that let's just look at 5 chromosomes, right? Let's say there's chromosome 1, chromosome 2, chromosome 3, chromosome 4, chromosome 5. So we have chromosome 1, let's say, is about this length, chromosome 2 is about this length, chromosome 3 is about this length, chromosome 4 is about this length, chromosome 5 is maybe like tiny like that. Okay, so we can see that these are all different chromosomes. But because we're diploid, we have 2 of each one. So times 2, times 2, times 2, times 2, times 2. So since we're just looking at 5 kinds of chromosomes, we know that there's another homologous chromosome for each one, so we're looking at 10 chromosomes total. And remember what homologous means. That doesn't mean that they're identical to each other in nucleotide structure. That just means that they have the same kinds of genes on them, but they have different alleles scattered throughout. So yeah, that's diploid. So there are 2 sets of them. Now let's look at something haploid. Haploid. I don't really know why hap is 1, to be honest, but I guess it just is. So haploid means there's only 1 set of DNA. Or yeah, one more like, I'm sorry, my bad. 1 set of chromosomes. So that means that if there's one about this length, there will not be another one about that length. There's just one. Cool. So you could have a tetraploid. That would be 4 sets of chromosomes. So there would be 4 homologous chromosomes for every 1 type of chromosome. But for now, we just want to worry about diploid and haploid. So diploid is 2 sets, and this will be pretty relevant moving forward. Before we start in this practice, I think that I did not explain how to find the ploidy the greatest, and I'm so sorry, and I love you. But so I think a good way to find ploidy would be how many homologous chromosomes are present for a given type of chromosome. So okay, let's just jump to the more difficult one. So we're looking at this big boy over here. So each one of these are a cell, and then they have a bunch of chromosomes in them. So over here, it looks like this is a pair of homologous chromosomes based on like the size of the thing, and this is a pair of homologous chromosomes, and this is another pair of homologous chromosomes. So there are 3 pairs of homologous chromosomes, right? But it's not a triploid, it is a diploid. And that's because going off of number of homologous chromosomes per chromosome type. So let's say that this is a type of chromosome, right? So there's only 2 of them. So that's diploid. Yeah. So the ploidy is diploid. There are 3 pairs of homologous chromosomes present, and there are 6 sister chromatids present. You can tell that it's in sister chromatid form by the X shape that it forms. Okay, let's check out this one over here. So there are no homologous chromosomes present. That means that there's just 1 homologous chromosome. It's not even a homologous chromosome, really, per chromosome type. So that is haploid. And we already know there were no homologous chromosomes present, but there were 2 sister chromatids present. And let me make a clarification. Both diploid and haploid and triploid and tetraploid cells have sister chromatids. Sister chromatids are a product of the DNA replication needed in order for the cell to replicate itself. So all cells need to replicate, right? So that means that all of them need to replicate their DNA beforehand, which means that they have sister chromatids. So let's look at this guy over here. So these look to be about the same length, and they're different colors. That means that they have the same genes, but they have different alleles. So there are homologous chromosomes present. So here's one over here. There's one over here, right? And there should all if you have a diploid organism, there should always be 2 pairs of everything, you know? So, but over here, number of homologous chromosomes per chromosome type. So this is a type of chromosome, right? And there are 2 homologous chromosomes present. It's diploid. And there are 2 homologous pairs present, and there were no sister chromatids present. We're looking for that X shape. Now over here, there are no homologous chromosomes because there are no other chromosomes that are about the same length, and so the diploidy would just be haploid. No homologous chromosomes, and no sister chromatids because they're not in that X shape. Alright, we're going to start talking about human reproduction. Humans are diploid, which means that they have 2 sets of DNA. You have a mom and a dad. You have 2 sets of DNA. Your mom is kind of a freak, so she has like 3, but no, she just has 2. And then your dad has 2 sets as well. So your mom and dad, they did it, and then they made you, and they gave you their DNA, and then your mom gave you half of your DNA, and your dad gave you half your DNA, right? But the thing is, they didn't give you all of their DNA, because if they did, then they would have given you 4 sets, which is not possible, because you have 2 sets of DNA, you know, and that would not, it wouldn't happen. So what had to happen first, was your mom had to create some kind of cell that had only 1 set of DNA, and your dad also had to create a cell that had 1 set of DNA as well, and then these came together to make you. Wow. So then now we're going to talk about gamete production. Wow. These cells that organisms create, or that humans create, for sexual reproduction are called gametes. They contain just 1 set of DNA. So they contain 1 set of DNA, that means that they're haploid. And they're also called sex cells. So for women, females, those would be egg cells, and for males, those would be sperms. So, each of these are haploid. They contain only 1 set of chromosomes, but it is a full set. So 1N, so a full set would mean 23 of them, 23 chromosomes. Sperms also 1N, 1 set of chromosomes, a full set of it, which would be 23 for humans. And then they combine to make you. So you, and then you have your diploid. So you have 2 sets of chromosomes for 46 total, because 1 set is 23 of them. So we need to talk a little bit about how a gamete is actually made. So, let's say the mom and then the dad. So the mom has 46 chromosomes. The dad also has 46 chromosomes. You have 46 chromosomes as well. So naturally, that means that the mom donates 23 of them, and the dad donates 23 of them, right? And, you know, like looking at your siblings, or at siblings in general, you know, they're not identical to one another. So, that kind of shows that the chromosomes that you inherit different ones. That the gametes receive random combinations of the chromosomes, right? But it's not as simple as just taking half of the 46 to get 23 and half of the 46 to get 23, right? So you are diploid. That means that you have so the human genome is composed of 23 kinds of chromosomes. Technically 24 once we get to sex chromosomes. Okay, never mind. I'm sorry. So we have chromosome 1, chromosome 2, chromosome 3. We'll just look at those for now, right? And you have 2 of each one times 2. So yeah. So you have 2 homologous chromosomes for each type of chromosome. So what will happen if we just take 23 of these at random, right? 23. That would probably result in so these are the creations of gamete. Let's say that you just put 23 into each gamete. That would result at complete random. that would for sure result in a bunch of them where there's there might be multiple of chromosome 1, so there might be 2 of them, right? You might have happened to get both of them, or you might chromosome 2, right? You may have a complete absence of chromosome 2. So what will happen is that when they combine, you might even get like 4 copies of chromosome 1. Let's say that this one, the gamete, happened to contain 2 of chromosome 1 as well. So then you would be tetraploid for chromosome 1. And let's say that the dad did produce 1C2 as normal. You would be haploid for chromosome 2. In fact, this would not really be a viable organism, and it would probably be miscarried by the body's systems. But yeah, so what needs to happen for gamete production is you need to get the gamete needs to contain one of each type, one and only one of each type of chromosome. Cool. So that means that each gamete will have chromosome 1, chromosome 2, chromosome 3, chromosome 4, chromosome 5, chromosome 6, chromosome 7, all the way to chromosome 23. And these are, I guess, chromosome 22. But and one of X or Y. Anyways, but it'll only contain one type of chromosome. But it needs to have at least one. It can't have more than one, and it can't have 0 of them, because then you would just be missing an entire chromosome. Okay. Let me think. And these are chosen at random. So let's go back to your parents. So let's look at just your mom. So your maternal grandfather and your maternal grandmother, each of them gave one set of chromosomes to your mother. But the thing is, you don't inherit that set unmodified, right? You don't just get the entirety of the set of your grandfather's chromosomes, They're chosen completely randomly. So you might ultimately get on your mother's side, you might ultimately get chromosome 1 from your on your maternal side, might be from your grandfather, one might be from your grandmother, whatnot. So gamete production is random. And only one of each kind. Meiosis is the actual process by which gametes are made. It goes from one diploid cell, so that has 2N, or 2 sets of chromosomes, to 4 unidentical haploid cells. So over here, unidentical. That kind of makes sense, because siblings, if all the haploid cells were identical to one another, siblings would just be the same. So each gamete has one of each kind of chromosome. So each gamete has 23 chromosomes. So that means gametes do not have homologous chromosomes. It's not possible. In order to have homologous chromosomes, you would need to have 2 of the same kind of chromosome, and that doesn't happen. So let's look over here. So meiosis, we're going to give a very brief overview, but it comes from a diploid cell. So this is a diploid cell. We can see by the presence of homologous chromosomes. They're about the same length, but they're different colors, showing that they're homologous. They have the same genes, but different alleles. And it looks like only 2 of the one type of chromosome is represented here, not the other 22 of them. And that's for simplicity, but it is happening for each one. So in order to go from 2N to like 2 sets of chromosomes to ultimately a sum of 4N, it looks like we needed 1 duplication over here in order to get there. So we do just that. If you remember, whenever we duplicate DNA, once we duplicate that chromosome, the resulting new chromosome, like the identical one, sticks to it, and that forms a sister chromatid. So that happens for each one. And then after that, homologous chromosomes line up. And this is where crossing over happens, which will be important in a second. So they'll line up in the center of the cell, and 2 of the chromosomes will interact. So these 2. And they line up like that. And this is happening all throughout for all the other chromosomes. They're lining up like that, like that, like that, like that. Yeah, but we're just going to look at these two over here. So they cross over, and during that crossing over, some of the nucleotides are swapped around where they interact. And this will be important down the line. So homologous chromosomes line up, and then they're separated into two cells. Okay, so, yeah. And where it crossed over, it literally just exchanged some DNA. Cool. So, looking over here, we separated the homologous chromosomes. These are now haploid. But the thing is, well, meiosis, we want to make 4 of them. Now we only have 2. But when your chromosomes combine, or when gametes combine, they aren't in sister chromatid form. They're just in that single chromosome form. So now we need the separation of the sister chromatids. So they'll line up in the middle. So at the moment, okay, so just to be sure, this one has 2N, so 2 sets of chromosomes. This one still has 2N. At the moment, each of these are only 1N. They only have 1 set of chromosomes, but they're not ready yet. So now we need to split them up again. Cool. So over here, let's say splits that way, splits that way. Okay. Cool. So now all of these are haploid, and they're not in sister chromatid form. And let's go back to this step. This step is very important. So this one's separation of sister chromatids. But this step over here, where the homologous chromosomes are separated, that means that each cell only has one of each kind of chromosome. So that ensures that the resulting gametes do not have any they aren't missing a chromosome type, or they aren't having an extra chromosome. So once you have these gametes, then they'll come together to make the full set. So these are 1N. After they combine with another gamete, another viable gamete, then they become 2N and they become diploid. And then they'll have 2 of each homologous chromosome for each kind of chromosome. A very important note that I don't really think I talked about, but whenever you have a separation of the chromosomes, so the first step with the homologous chromosome separation, and the second step with the separation of the sister chromatids, those are completely random. So whether it goes into that cell or the other cell is completely independent of the other ones. So a crazy amount of combinations are possible. And the crossing over, so creating new kinds of chromosomes, also contributes to that genetic diversity. We gotta cover 2 of Mendel's laws. I'm going to start with Mendel's law of segregation, which states that 2 alleles of a given trait will separate randomly from each other into gametes. So this basically describes meiosis. So separate. So this is the starting diploid cell. Let's say that we're only just looking at chromosome 1, or that we're just looking at chromosome 1. And let's say that at this given location, that's the gene for hair color. This chromosome has the red hair allele. This chromosome has the black hair allele. So what's going to happen is that during meiosis, they're going to separate from one another. That's the separation of homologous chromosomes. And they're each going to enter a different cell. And that cell will eventually form the gamete. And there was a completely random chance of it, right? There was a 50% chance of this entering a particular one, 50% chance of this entering a particular one. So they're separated, meaning that your haploid cells will not have 2 of the same kind of chromosome. It's haploid, and it was random, so it was 50-50 whether one gamete would have this one or one gamete will have this one. So that means that half of all the gametes produced will have the red hair allele, and half of all the gametes produced will have the black hair allele. So we've been seems like we've been using alleles and chromosomes kind of interchangeably, but think about it this way: each of these are a chromosome, and they have alleles. So if you inherit one chromosome, you get the allele. So if there's a 50-50 chance of having this chromosome, that means that there's a 50-50 chance of getting the allele that is attached to it. So that's just Mendel's law of segregation. So to reiterate, we want to focus on two parts of this, and the separate part and the random part. So each gamete, oh my, has one allele of a gene. And separation of alleles is random. Second law is the law of independent assortment. Alleles separate/sort independently of other alleles, i.e., the assortment of one allele into a gamete does not affect the assortment of another. So let's take a look at these two traits. So the C is for cystic fibrosis and the H is for Huntington's disease. So where the big H is disease, so Huntington's, and the little h is healthy. This isn't really important, but whatever. So the big C is healthy, and in this case, the little c would be cystic fibrosis. Okay, so the law of independent assortment says that, okay, well, let's think about the law of segregation. So this big C and his little c are segregated, right? So half of the gametes will have a big C, and half of the other gametes will have a little c. So it was 50-50, the chance of this one having either one. So the law of independent assortment says that, okay, I think a better example to think about, or a better way to think about it, would be if it was not independent. So it was dependent assortment. Let's say that this big C and this big H are like linked together. So if you have a big C, that big H will follow it. Do. And if you have a little c, that little h will follow it. So that would ultimately result in alleles not being independent of one another. Where if you get one allele, there's a much higher chance of getting the other allele. But that does not actually happen. It's 50-50. So this big C could either get this big H, or this gamete, if this big C is there, that doesn't really matter. It could be either the big H or the little h. So that's the law of independent assortment. It increases genetic variability. And okay, you really don't need to know this, so don't think about it too much. But the law of independent assortment usually checks out because of the crossing over of the homologous chromosomes that happens during meiosis. But in actuality, there are quite a few alleles that are linked together. So if you get one allele, there's a high chance that you'll get the other allele as well. But for the purposes of this class, the law of independent assortment holds. The actual biological mechanisms of gene expression, and that will also explain why an allele is said to be dominant or why one might be said to be recessive. The main thing I want you to take from this is that the interaction between the genotype and the environment produces the phenotype, which is the manifestation of gene product interactions. Okay, so what does it mean? What is a gene product? So we have our gene, it codes to a polypeptide. We would have to transcribe our gene, and then we would have to translate it. And then from that, we get our gene product. And it is exactly what it sounds like. It is a product of our gene. And the reason why we use the word gene product is because we may or may not actually get the functioning protein based on what we have in that gene over there. So we might have our good functioning protein, or that gene might be mutated, and then we might have a loss of function protein. meaning is a polypeptide that does not really do what it's supposed to anymore. So within a given organism, its genes, or within a given cell, its genes are expressed, and that produces the gene product. Now, there are a lot of different factors that come into whether or not a gene is actually expressed, and I guess we can talk about this. But everything within the human body, like all the cells, contain our full set of DNA. So a skin cell has the same amount of DNA as something found within our kidney cell, the same DNA. However, the DNA from that are regulated differently. Different proteins are expressed in our skin cell compared to within the kidney cell. So that allows differentiation to happen. So yeah, and that is because they have different gene products. So for diploid organisms, and that means having two sets of chromosomes for each one. So they have a bunch of homologous chromosomes. So that would be the presence of homologous chromosomes. And this should really be cells, not really organisms. Homologous chromosomes. And then that would be two sets of chromosomes. This could happen twice. Because we have two homologous chromosomes, that means we have two possible alleles because they share the same kind of gene type. So each allele could be expressed and produce their own gene product. So each allele can be transcribed and translated to produce that gene product, and that would happen twice for each organism or for each gene. For a haploid organism, that would just happen one time because they only have one chromosome for each. They only have one chromosome, which means they only have one set of genes possible. Okay, so let's go back to over here. So the interaction between genotype and environment. So there are a lot of different things in play that can actually determine genotype expression. It could be, I don't know, nutrition, even altitude. I don't know, just like a bunch of different things that can influence whether or not our genotype is actually expressed. And that produces the phenotype, and then that phenotype is the result of the gene product interactions. And what do I mean by gene product interactions? Well, because the diploid organisms can potentially have two competing gene products, they can counteract each other, they can work together, and that would produce a different phenotype. We're going to talk about gene product interactions of diploid organisms. This is stated on the study guide, but it won't be on the exam. So maybe just sit back and chill out because I think that it is useful to learn conceptually. Cool. So let's say that we have a haploid cell or a haploid organism. So that means that it only has one set of each chromosome. So this is a chromosome, there's a gene there, there's a gene there, there's a gene there. So for each gene, the gene product that it produces will form whatever phenotype is associated with it. But for a diploid, we do have two sets, like humans, we do have two sets of chromosomes, and these are homologous. So that means that we have two genes over here that code for the same kind of thing, but they can be different from one another. So what happens is that you have the potential to get two different gene products, so two different polypeptides most likely, and they can interfere with one another. And that is the interactions we're talking about. So just because you have one thing that codes for a trait doesn't necessarily mean that the trait will actually be observable because of confliction from the other one. So there are three situations that are over here. We're looking at a flower, and we're looking at the color of the petals. So let's say that the genotype for each one is big R, little r for their petal color. So our big R allele is said to be code for red, and the little r is said to produce the phenotype of white petals. Okay, so looking at this example, we see that the petals, even though each one of them are heterozygous, meaning you don't have like two dominants and two recessives, that this one is completely red. We don't see any hint of white in there. So this is a case of classic dominance.
what could be a biological explanation for this? Well, okay, well, to review, with diploid organisms, both alleles can be expressed. They can be transcribed, and then they can be translated by the ribosome, and they both can produce a gene product. But let's say that when the big R allele is red, it produces the red pigment. It's a protein that causes the red coloration. But what happens when the little r allele is red? This one might have been mutated down the line, so it is dysfunctional now. So it might be we just produce a polypeptide, but it is no longer a white pigment. It doesn't really do anything. It's still made, but it's not really like a pigment protein anymore. It's just like there. So what happens is that there is only one kind of protein that can influence the color, and by default, that is just red. Now, let's say that we had the little r, like a homozygous recessive phenotype. We would see that there are only white petals. But why might that be the case when the little r ultimately codes for a dysfunctional protein? It could be that the petals are naturally white without the pigment. So when you don't have anything at all to influence the pigment, it's just white. Now, you can also have a dysfunctional allele where instead of there being a mutation within the coding region such that it results in a different protein, you might not even have the promoter. So remember, the promoter is what it allows transcription to happen. And if you had a mutation that deleted the promoter, then you just wouldn't produce any gene product. So you would still only be producing that red, and then that red is the only one there by default. In this case over here, we have pink, and we know that pink is a blend between red and white. So this is an example of incomplete dominance. We can see that neither one is able to distinctly show the phenotype associated with it. So we still have the genotype big R, little r. A biological mechanism that might explain this would be big R produces the red pigment, a protein red pigment, and the little r produces a white protein pigment. So over here, in this example, I gave a loss of function protein where it didn't even code for a pigment, but this one does code for a pigment. So if you have, by nature of the petals, if you have red and white and they're all spread out, then like the proteins are very small. And then when you look at it from like the naked eye, it's going to look pink, and that would be its phenotype. Neither one of them are dominant. Both of them are there, but like they can't like overcloud each other. And then over here, this is an example of co-dominance. So the red was dominant over here, or the big R was dominant over here, and we can see distinctly the phenotype it produces. These are co-dominant because we can distinctly see the phenotypes of both of them. We can see red and white. So this means that, well, okay, what would be a biological mechanism for this? So like R and R still produces that red pigment, and little r also produces that white pigment. But for whatever reason, they don't really mix, so you can distinctly see the phenotype of each one.
All right, we're going to talk about the difference between autosomal and sex chromosomes. I've critically referred to it a couple of times, and it's a very interesting concept. But we're not going to get too much into the details, but if you notice a gap in your knowledge and you would like to know more, please feel free to email me. I'd love to talk about it. So autosomal chromosomes, those are chromosomes 1 to 22. Sex chromosomes are that 23rd chromosome, and they are special. They determine your sex characteristics. So we have X and we have Y. Those are the two kinds that you can have. And where all the autosomal ones, you have homologous chromosomes present for each one. So you have, for sex chromosomes, you might not have homologous chromosomes present. In fact, only females have homologous sex chromosomes. So the sex chromosomes, they have genes that correspond to sexual dimorphism. So it's what makes males males and females females. So on the Y chromosome, there are different genes on there that help produce the development of the testes, for example. And the presence of two X's would determine the present like female genitalia, whatnot. So the females have two X chromosomes. So these are homologous to one another. The males have one X and one Y, and these are not homologous. So The X chromosome is pretty normal, it's pretty healthy, but the Y is like a very like shriveled up little thing. It's not really homologous. It's kind of, but whatever. We're getting into the nitty-gritty. I'm sorry. So yeah, females XX, males XY. So let's look at the creations. So let's say that these two were to have a kid, and let's start with daughters first. So the daughter as a female has XX, and while they aren't homologous, the male cannot donate both of them. They only donate one or the other one, same as a female will only donate one or the other one, and this is the law of segregation. So the daughter has to get one X from the mother, and it could be either the one from the grandfather or the one from the grandmother, so the maternal one or the paternal one. So, paternal, patriarchy, okay, paternal. So, yeah, so the daughter gets one from the mother, and it could be either this one or this one, but she also gets one from the father. So the father can only give one kind of chromosome. I mean, one, yeah, it just is an X. So whatever the X the father has is what the daughter will have, that X as well. And then let's look at the son. So the son as a male is XY. So again, the same thing. So the son can get either X from the mother, 50-50 chance, but he can only get the Y from the father. So if the father has that Y, the son will inherit the same Y. Cool. Okay, so yeah, this has a lot of different implications, particularly on diseases. So we're going to talk about an X-linked disease and a Y-linked. Okay, so X-linked, that means that there is a disease that is present on the X chromosome. Okay, so let's think about a daughter and let's think about a son. Okay, so if there was an X-linked disease, what'll happen is that males would be disproportionately affected. So by probability. So let's think about it. Let's say that we're looking at a gene that codes for a very important protein carrier. And that when you have the recessive variant of the gene, the recessive allele, it has a broken promoter, so you don't even produce a gene product. So for a female can have two alleles of that gene. So she might inherit an X with that recessive allele, meaning that no channel protein is made for that, but she might also have a healthy one. And that healthy chromosome will produce the channel protein, and that'll be enough to provide the healthy phenotype. But for the males, the sons, if they get just one copy of that recessive X on recessive allele on their X, they're screwed. So they don't have another X with a corresponding gene that could potentially provide a buffer. You know, you get one, you're not good. You know, whereas in order for a female to get that unhealthy one, she would need two copies of the recessive one, whereas you just need one because you don't have anything to help counteract it. Okay, and next we're going to talk about Y-linked. So this is a disease on the Y chromosome. So it would be impossible for women to have it because they have XX, so they cannot even have the Y. So that means that if there is a Y-linked disease, and because the son can only, so this is the mom, the dad, and this is the son. So the son had to get a Y chromosome, and only the father could have provided it. And then by the law of independent, law of segregation, the mother would provide one of the X's, and it can be either one of these. So that's it. So if the father has a gene on that Y that is a disease, the son will have it as well, because the son inherits the same Y. So Y-linked sons are disproportionately affected. And we'll also be talking about different stuff like X-linked recessive, X-linked dominant. So just real quickly, let's think about X-linked recessive. So that would mean that for a female, in order to have that phenotype of that of the recessive allele, she would need 2 of them, where a male would only need 1 of them because there's no other X to counter it out. But let's think about X-linked dominant. That means that if you have that allele, it'll overshadow other alleles. So for a female, you would only need 1 of those to get it, and for a male, it's kind of the same thing. You just need 1. Right. Let me think. Y-linked. Cool. And it doesn't really exist for Y-linked dominant or Y-linked recessive, because if you just have 1 Y, you have it either way. There's nothing to compare it to. Okay, I think we finally covered all the conceptual stuff, and now we're going to talk about some problem-solving stuff. Yay. So, gamete determination. That is the difficult part. So, you can have 2 different ways of doing it. They might give you a visual one, like that, and then you'll have, like, different genes popping out, right? Like that. Or they might just give you the genotype, and then you have to make the gametes for that. That one's pretty common. So, So let's say that you're only just looking at 1 gene, right? So, this is the genome for the mom, and this is the genotype for the dad. So that means that 50% of them will have big A, and then 50% of them will have little a. And then for the dad, all of them will have B big A. And then this is where you make that Punnett square. So the Punnett square, after you determine the gametes, the Punnett square serves to combine them and then show you which genotype the offspring will have, or the probability of it. So we'll do the basic 2x2 one, right? So let's look at the maternal one. You can either have big A in one of them, and little a in one of them, 50%, and then all of them 100%, right? So over here, you cross them. Okay, it looks like 50% of the there's a 50% chance of the offspring being homozygous dominant, and a 50% chance of it being heterozygous. Okay, so yeah, now we're gonna talk about looking at more than just one trait, more than 1 gene. So let's see, we're looking at this. Okay, this is the maternal one, and this is the father. So now, so remember the law of independent assortment? I mean, the law of segregation. So each produced gamete needs to have 1 of each kind of gene, 1 of each kind of allele, right? So if there is an A allele, that has to be with a B allele. So here's the formula that we can use. Oh, wow, this is not very well. So you cross this one over, and then you do that, and you do this, and you do that. So we'll do the exact same thing over here, right? So over here, you can make a B. Over here, you can make a little b. Over here, you can make little a, big B. And then over here, you can make little a, little b. So we'll do the same thing over here. You see that because he's homozygous for both of them, it would just be A, B for all of them. Cool. So now we can do a cross, and this one's pretty annoying, or it's more annoying. So it is the 4x4. So remember, we have the gametes at the top, and each section is a gamete. So there'll be gamete over here, gamete over here, gamete over here, gamete over here, and then we cross them, we combine them, just like sex. Wow, it's like we're having sex. Haha. So anyway, so we combine the gametes, and then we see the genotypes of the offspring. So this is one gamete, AB, another gamete, AB, I mean they're all AB, and all of these would be a little b. Cool. So A, B, 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 yeah. You're just crossing over like that. So those are Punnett squares. And whenever you're solving these, you need to keep in mind which combinations will produce what kind of phenotype, you know? Will it produce, is there something dominant? What's recessive to what? And now we're going to talk about basic probability. And we're going to go over some practice problems, and those will help a lot. So we're just going to go over some basic probability. So whenever anything asks you and, so it's this and this, you multiply. Whenever they ask you or, you add. Okay, so you're given a question. What is the probability of having a boy? Okay, we'll do it. Okay, just real quickly. Okay, so big B is black hair color. Little b is blonde. So this is, it doesn't even matter which one's the mom, which is dad. But you're asking, what is the chance that this couple will have a boy that has blonde hair? But this is an and question, so it's a boy and blonde hair. Okay, so we gotta think about a couple of things, right? Well, we gotta think about which genotype would give the phenotype for a boy, and there's only one, and that's XY. And which phenotypes would give it for blonde? That would be this one. That's the only thing, because we know that the little b is recessive. Now let's say that it asked you what kind of genotypes. What would be the probability of getting black hair, right? And we were trying to determine the genotypes possible. It could be either big B or big B, big B. But for the recessive one, that's the only option. It has to be homozygous. So, yeah. Blonde. Is there an E at the end? No, there's not. Oof. So, okay. We could make the 4x4 one, or we can make two separate Punnett squares. I personally prefer that one. So let's start with the boy first. So we know the mother's genotype is XX. We know the father's is XY. XX, XX, XY, XY. So there is, looking over here, there is a 50% chance of having a boy. XY, and that makes sense. So there's a 1/2 chance of having a boy. And then for blonde, let's do this crossover here. B, B, B. So looks like that half of the crossing overs will produce a homozygous recessive for the little b allele, which means that half of them will display the phenotype for blonde, right? But this is the and question, so now we multiply them. So 1/2 times 1/2. Okay, there is a 25% chance that this couple will have a boy that is both blonde and that is a boy and blonde. So, it might ask you three things, like, what is the chance of You can just say three separate Punnett squares and then multiply them together. Now, let's say that you had an OR question. What is the probability of getting a boy or a girl that is blonde? So, now we add them together. We find the probability of each and then we add. So, it's the same thing because it's 1/2. So let's look at a boy and then a girl, or a son and a daughter. Okay, so and they're blonde. So there's a 1/2 chance of getting a boy, 1/2 chance of being blonde. We multiply them together. That is a 1/4 chance. There is a 1/2 chance of getting XX, that's a girl. 1/2 chance of getting little b, that's a blonde. So 1/4. And now we add them together. So there is a 1/2 chance of getting a boy or a girl with blonde hair. So that's basic probability. Alright, Rooney. Let's look at a pedigree. I downloaded this PDF from Google like 2 minutes ago and I have not looked at it yet. So let's look at the pedigree in general. So whenever you have horizontal lines connecting them, that means that they mated. And then those things going off it are their offspring. So whenever it's dark, that means that that individual has the phenotype for whatever trait you're looking at. Pedigrees are usually looking at diseases, so it looks like this one over here, this one over here, this one over here have the phenotype for the disease. The circles are the mother, and the squares are the father. Or, I mean, not the father. So circles are females, squares are males. So that's important when you're trying to think about, is something X-linked or is it Y-linked? So is this trait dominant or recessive? So let's take a look over here. Okay, it looks like of these 5 children, 2 of them have the disease. But looking over here, 3 and 4, neither the mom or the dad have the phenotype for the disease. That means that they carried it. So that means that it is recessive. So over here, well, let's think about it, right? Let's say that we're looking, let's call it A, right? So let's say that we were like this. Well, then these would have the disease because they have the 2 little a's, right? But let's say that it was dominant. If it was dominant, it would either be that, or that, or that. But that would mean that these 3 and 4 would have that phenotype of disease. So that's not really possible. So they're actually just carriers. Little a over here, And what happened is that when they crossed, some of the children happened to get both of the recessive alleles, so then D3 and 4 have disease. So that's recessive. Yeah. And it was only possible if they were heterozygous, because they didn't have the trait and the phenotype of the disease, but they still spread it on somehow. Okay, yeah.
Example question 3. Hi, please tell me the ploidy and if homologous chromosomes and or sister chromatids are present in the cell to the right. Thanks, Justin 1. One time I sent an email to someone, it was my boss, and I spelled my last name wrong. That kind of sucked. Anyway, okay, so the ploidy. So that's asking, is it haploid or is it diploid? So we can tell ploidy by the presence of homologous chromosomes. And we see that there are homologous chromosomes. There are 2 sets of homologous chromosomes, these and these. So because there are 2 sets, that would be a diploid. Okay, now we know we just said that they were homologous, but why are they homologous chromosomes? Well, homologous chromosomes are chromosomes that are the same type of chromosome, but they are ultimately, they have different alleles on them. So one from the mom, one from the dad, and so in this case, yeah, it does have homologous chromosomes. So yes, homologous chromosomes. And sister chromatids are sets of chromosomes that are replicated and duplicated, and then they stick together. So we do have that. These are identical to each other. They have the same alleles as opposed to homologous ones. So yes, we do have sister chromatids. Example question 4. Rat sub 1 and rat sub 2 are clones that differ only in their sex chromosomes. They have gray fur. They mate a lot and produce the following progeny. Assuming fur color is not coded within the sex chromosomes, what conclusion could be drawn about the dominance or recessiveness of fur color? Okay, so we're given this. So when you're given a problem like this and you're given all their progeny, you want to find out the ratio. So there are 15, 28 plus 472. That is 2,000. Okay, so 1528 divided by 2,000, and then 472 divided by 2,000. Okay, you could probably estimate that that's about 3/4, and the other one's about 1/4. But let's get the exact number for fun. And also, you wouldn't need a calculator in an exam. Okay, so our proportion over here is 0.764, and this one is 0.236. So very close to 3/4 and very close to 1/4. Now we know that rat sub 1 and rat sub 2 are that they have gray fur, and that means that, and because they're clones, they share the same kind of genotype for their fur color. And because they are not only producing gray, we know that they have to have a white allele in the background. So that means their genotype would be, let's say that for we do F, right? Where big F, like that, because they have to have some kind of white one in the back. So let's say that the big F is gray, and this is our Punnett square, and the little f is white fur. So we can do FF, 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 and FF. Okay, so over here if big F was dominant, we would expect 3/4 of the progeny to have gray fur, and we would only expect 1/4 to have white fur. And that is very similar to the proportions that we have here. In the real world, it's not perfect, but the more you do something, kind of like flipping a coin. If you flip it 10 times, there's a pretty low chance that you'll actually get the 50-50, even though you know it's fair. But if you flip it 1,000,000 times, you'll get very close to the proportion of 50-50. But in this particular question, if you are able to do this one, that will be maybe helpful. Haha. So you might have noticed in your practice exams and in your other resources that on most questions you're given a model. So on the test, it would be reasonable to also be given a model as well. You probably would not be given just the genotype, So example question 5. Given the following homologous chromosomes, so this is one set of homologous chromosomes, this is the other set, and that recombination is not possible, so you don't have any crossover happening over here, what are the possible gamete genotypes? So that means that our gametes, because we have no crossing over, they either have this one, or they have this one and this one, or they have this one and this one, or we have this one and this one, or this one and this one, this one. So it's just a probability game. So because this is haploid, we always oopsie daisies, because we are ultimately trying to get a haploid genotype, and by law of segregation, we don't have two homologous chromosomes in the same haploid thing. We have A is always paired with big G, or big A is always paired with big G, and we can have this one. We can have AGD, or we could have AGD, because we could have had this one by chance as well, law of segregation, and then we do the exact same thing with this one. We have little a always comes with little g, and that would go with big D, or it could, and then we have AG little d. Cool. Example question 6. Given the following homologous chromosomes, so that would be this pair over here, and that crossover is possible, and also another word for crossover is recombination. Recombination. What are the possible genotypes? So hypothetically, let's say that crossover was not possible. That means that our possible gametes would only be AF and only little a, little f. So those are our only possible gametes, because because we can't have two homologous chromosomes in the same gamete. So we already have one and the other one, and that's about it. But crossover does happen, and crossover happens for the same allele. So basically, what happens is that crossover between two homologous chromosomes, they happen at the same location, and this location will be like a gene. So they trade genes. So one chromosome will give its gene, will trade. So it's like swap, So in this case, you could swap. Oh, well, let's think about it. You might not even have any crossovers by chance. So you have big A, little big F. You keep that. You have little a, little f. And yeah, but let's say that you cross this one over. You trade your A on this neon pink one to this light pink one, and the light pink one gives its little a in return. So then after the crossover, now you could have. Sorry, I don't know why I'm so confused. I think it's because I'm tired. So you have big A, little big F, little a, big F. Oh no. And then you can have big A, little f. And you could consider the possibility of this crossing over right here, but you'll notice that if we do that, we'll get big A, little f, and we already did that. And then we would get little a, big F, and we already did that as well. So we're good. Cool beans. Example question 7. You have homologous chromosomes. It's crossing over. Yeah, let's do it. So okay, it doesn't matter if we cross over here because we're only focusing on 2 genes. Doesn't matter. But here it does matter. So well, let's get the genotypes of the current state that we have right now, because it could also not cross over, or you might have a double crossover, which will result in the same kind of thing. So you have T, and then you have big Q, and then you can have A, or you can have T, big Q, little a. And then over here, you could have T, little q, big A, T, little q, big A. Okay, let's say that you have a crossover going this way. So this results in this big T now being a little t, and this little t now being a big T. Okay, now what we got? We have T, we don't have that combination yet. We have TQA. Okay, yeah. And then we would also have TQ little a. And over here, little t, little q, we don't have that combination yet. Little TQA, little TQA. Cool. So I do a lot of these problems visually, and yeah, don't want to overcomplicate stuff. Oh, this one's quite the hoot, and that is example question 8. So I kind of simplified it down a lot, but oftentimes diseases are recessive, which means that in order to get the phenotype, you need to get 2 recessive alleles for it. And that if you have a dominant allele, you'll just be a carrier for it because you have that recessive in the back, but you express the healthy phenotype. So in this case, little a will give you asthma, little d, type 1 diabetes, little l, small lungs. Okay, yeah. It doesn't mean that if you had a big allele for each one, a dominant one, you would have the healthy wild type phenotype. So we're given the genotypes on the mom and dad. The mom has little a, she has asthma, and she is a carrier for the rest of the stuff. A carrier means that you don't have the phenotype of disease, but you carry the potential to spread it onto your offspring, like that. The dad, looks like the dad has small lungs. Oof. But he has the phenotype big A, big D, and then little a. Cool. Okay, so what is the chance of everything normal, but the kid has asthma? Okay, so looking at, you know what, it's simple to just do, okay, so basically, let's break it down. Everything normal, so that means that we need at least one D in there. We need at least one big L in there, but we need little a. Okay, and we can do 3 Punnett squares for this. So let's look at the mom genotype. That would be AA, and the dad genotype would be big A, little a. Because this is what we're looking for, we see that there is a 50% chance of AA, which corresponds to a 1/2 chance. So let's start off with that 1/2. And then whenever you're looking for the probability of like multiple things happening, usually is fair game, just multiply them together. So now we're looking for at least one D. Well, we'll just cross them over now. There's a big D, D, big D, big D, big D, big D, little d, big D, little d. Okay, we see that there is a 100% chance of having the phenotype of because the dominant one, you see there's a 100% chance of having no diabetes. So that would be ×1, or like 1/1. And then now lastly, the lungs. Let's cross them. So then we have the mom. Let's put her on top. LL, and then we have little l, yeah, big L, little l, big L, little l, little l, little l, little l, little l. Okay, so looking at this, we see that 50% will give us that this, which means that while you're still a carrier, the phenotype you actually express, you do not have small lungs. So you have the normal wild type. So that is a 50% 1/2. That's 1/2. So multiply everything together to get the chance of all 3 of them. 1/2 times 1 is 1/2. 1/2 times 1/2 is 1/4. So there is a 1/4 chance that that resulting kid is everything normal, but they have asthma. Okay, what is the chance of asthma and small lungs? So that means that you don't have type 1 diabetes. So that would mean that you the genotype we're looking for would be AA, and I'm going to write it a little bit differently than I did here. So in small lungs, we would have LL because we need 2 with the recessive ones, and we do not have type 1 diabetes, which means we either have a genotype big D, big D, or big D, little d. Okay, so we're looking for this. So we already did the crossover here. We're looking for the same thing, like little a. So we have a 1/2 chance of getting asthma, and then little l. We did that over here too. There is a 1/2 chance of getting asthma. Times 1/2. And we also did diabetes one. Huh, how about that? And there is a 100% chance that you will not have diabetes, which is yeah, okay. That would be 1/4 as well. Cool. Okay, chance of all the bad things. So you have asthma, you have type 1 diabetes, and cherry on top, you have small lungs. Oh man. So chance of all the bad things. Okay, asthma, you already know that's 1/2. And oh, asthma would be like the little a. And then the chance of small lungs, that would be, no, actually, let's say chance, we're going in order, right? That would make sense. So we're looking at the chance of diabetes, the phenotype for type 1 diabetes. And we see, no, you're actually covered for all of them because you have that dominant D for every single one. There is a 0 chance. So that would be 0 divided by, we'll do 7. Ooh, keep it spicy. And then, there's no point in doing the little l, but we will. So what's the chance of getting a little l? We go back to our Punnett square. We see that there is a 50% chance. So that'll be 1/2. 1/2 times 0 is 0, and 0 times 1/2 is still 0. So 0. Oh wow. Cool.
So we're gonna do the genetic sample questions with explanations. If you guys haven't looked at the questions before, I recommend stopping and trying to do it yourselves first before watching me do them. So a diploid organism with 2n equals to 46 has how many sister chromatids in total? So we know one chromosome consists of 2 sister chromatids. This is one chromosome, and we have 2 sister chromatids in one chromosome. So this question is asking, we have 46 chromosomes in total. 2n equals to 46 means we have 46 chromosomes in total. And each chromosome has 2 sister chromatids, right? So for this, we're going to take 46, our chromosome number, and we're going to multiply it with 2, the number of sister chromatids each chromosome has. And then we're going to find that we have 92 sister chromatids in total. So for practice, I put these. Sometimes you get pictures, sometimes you get numbers. So with this, you're going to count the chromosome numbers. So 1, 2, 3, 4. We have 4 chromosomes in total. We know each chromosome has 2 sister chromatids. So we're going to multiply 4 with 2, and we're going to get 8. Similarly here, we have 1, 2, 3, 4, 5, 6 chromosomes in total. And we're going to multiply that with 2 because we're looking for sister chromatids. And we're going to get 12 sister chromatids in total. For second question, will mitosis or meiosis result in more diversity? And the answer is meiosis. And that's because of crossing over. Crossing over, this is from lecture 2. Crossing over increases genetic variation, and how that happens is when chromosomes swap alleles, like here, we swap the C allele, and here we have this chromosome has the genetic material of this, and this one has the genetic material of this, and that's how we create new combinations of alleles and how we increase genetic variation. So the next question is about incomplete dominance, and incomplete dominance is basically the offspring not looking like both parents. So let's say we have a red flower here, we have a white flower here, and the offspring looks pink, which is not like both of them. And this is what we call incomplete dominance. So the heterozygote offspring is going to be the intermediate, and that's incomplete dominance. So this question is asking, a gardener is trying to estimate how many purple roses he could possibly get from crossbreeding his blue and red roses, assuming incomplete dominance. So we're gonna find the genotypic and phenotypic ratio of the offspring in F1 generation, and then we're gonna self-cross F1 and get F2. So for that, we know one of our parents is big R, big R. So we're gonna put big R here in our Punnett square. And then we have little r, little r. So we're gonna put little r, and then to cross them, we're just gonna do big R, little r, and put it here. And then we're gonna do big R, little r, put it here. Similarly, big R, little r, put it here, and big R, little r, put it here. So here we have 100% big R, little r, and we go up and see big R, little r is purple. Here, I also put them here. So that means our genotype is going to be big R, little r, and our phenotype, what we look, is going to be purple. And because we have 100%, we're just going to have genotypic ratio of 100% big R, little r, and we're going to have the phenotypic ratio of 100% purple. So since we have 100% purple, we're just going to take big R, little r's in both and cross them. So it's going to be big R, little r here, big R, little r here. And then we're going to start crossing them. We're going to have big R, put them here. Big R, little r, put it here. Big R, little r, and here. Little r, and put it here. So when we see this, we're going to have 1 big R, 1, 2 big R, little r's, and 1 little r. So we're going to have 1, 2, 1 ratio. And similarly, because it's a heterozygote, our phenotype is going to look different. So we're going to have 1 blue, 2 purples, and 1 red. And that's why we're going to have 1 to 2 to 1 for our genotypic and phenotypic ratio. Okay. This question is asking, in a species where a round shape, big W is dominant to the wrinkle shape, and that's recessive, what are the genotypes and phenotypes of offspring in the F1 from a cross between a wrinkled and round pea plant? And we have complete dominance. So it's going to be like how we always do. So wrinkled is going to be our little w, and the round one is going to be big W. And then we're gonna cross them, big W, little w, and put it here. Big W, little w here, big one and little one, and here. So we see we have 100% big W, little w. We have 100% heterozygotes. And the phenotypic ratio, because this is complete dominance, we're gonna have big W, little w, right? So we're gonna see that round is dominant over wrinkled. So we're gonna have 100% round in phenotypes, okay? So this question is about X-linked genes. It's asking, red-green color blindness is an X-linked recessive genetic disease. If Amir has a red-green color blindness and his wife Fatima has no history of this disease in her family, what's the probability that their sons will inherit this disease, and what's the probability of their daughters inherit the disease? So for this, we're gonna think that Fatima's entire family got tested and they found no color blindness. So we're gonna, and because it's a recessive disease, and generally, like when a family has no history, we normally assume that they're gonna be homozygous for the trait, for the dominant trait. So we're gonna be saying that Fatima is gonna have X big R, X big R. Okay. And then the father has color blindness. So he's gonna have 1X because males have 1X and 1Y. And the 1X that he has is gonna have a little r. Okay. So we're gonna cross them. We're gonna put X little r and Y here. We're gonna put, this is the father and this is the mother. We're gonna put X big R, X big R, and then we're gonna cross them. So it's gonna be big R, little r, big R, little r, big RY, big R, little r, big RY, and put it here. So we're gonna see the probability that their sons inherit the disease is, we're gonna look at the ones that are male, and we're gonna see they're both homozygous dominant. So they're both not going to be carrying, not going to be colorblind. And we're gonna say they're 0% colorblind. And then for the daughters, we're gonna check here, and we're gonna see that daughters, none of them will have color blindness, will be colorblind, but they're going to be carriers because we have one dominant and one recessive. And that's why we're gonna write 50%. Nope. Should be 100%. Should be 100%. Ignore this. It's going to be, they're going to be carriers. Ignore this. So both crosses are giving carriers, and that's why we're gonna say it's 100% carriers, and the daughters will inherit the disease but not show the trait. Okay. So for this question, we're gonna list all possible gametes with big A, big B, little b, and we're gonna cross them like A, and we're gonna go like this. Okay. So big A, little b, sorry, big A, big B here, big A, little b, we're gonna write it here, little a, big B here, little a, little b here. So we're gonna have 4. And to check this, we have an easy formula. This only works for heterozygotes. It does not work for anything that's homozygous, okay? So because this is heterozygote, they're both heterozygotes, we're gonna use this formula. It's 2 to the n, and n is going to be the number of letters we see, okay? So we see A, right? So it's gonna be 1, and we see B, so it's gonna be 2. So for this, we're gonna have 2 to the 2, 2 to the second is gonna be 4. And that's what we got. We got 4. So we're fine. And that's how we check. But it only works if all of them are heterozygous, okay? So here we have big A, big B, little b, big C, little c. So we're gonna cross them. A, big B, big C, big A, little b, big C, big A, little b, little c, big A, big B, little c, little a, big B, big C, little a, big B, little c, little a, little b, big C, and little a, little b, and little c. So I have 8 here, and I want to check this. I see we have heterozygous, heterozygous, heterozygous. So we can use this formula. So we're going to use it. So we're going to have 2 to the third, and that's going to be 8. And we're going to count 1, 2, 3, 4, 5, 6, 7, 8. I have 8, so I'm fine. This is correct. And then for this one, we can't use this formula. And that's because we have here big A, and here big A, big C, big C. So for this, we're going to check big A, big B, big C, and big A, little b, and big C. And we can't find any other combination. And that's because we have 2 big A's and 2 big C's. Okay. So for this question, we're going to perform a dihybrid cross between 2 plants with genotypes big T, little h, and little t, big H, little h. And here we're going to assume complete dominance, and we're going to try to find genotypic and phenotypic frequencies. So for this, first of all, we try to find the possible gametes. So for this one, we're going to put it here. We're going to see we have 1 big T, 1 little t, and that's it. We don't have any other possible gametes for this one, right? We don't have any other possible gamete options. Okay, and then this one is going to be little t, big H, and little t, little h. So now we're going to try crossing them. We're going to put this up here. We're going to put this here and this one here. So when we cross them, we're going to be seeing big T, little t, I'll put it here, big H, little h, put it here. So this is 1. And then we have big T, little t, put it here, little h here. So we're going to have 2 different genotypes, right? So we're going to have 1 T, the big T, big H, little h, 2, 1 big T, little h, little h. So it's going to be 1-to-1 ratio. Or you can write it as 50% this, 50% of that. And then for phenotypes, we're going to have still 1-to-1. And that's because this one, 50% is going to be tall with red flowers, right? Because this one, we have tall red flowers because it's heterozygous and it's complete dominance. So we assume that it's tall and red. And then the 50% here is going to be tall with no flowers. So it's going to be 1-to-1 ratio. For this question, we're going to look at blood types. So the question is asking, Michael's blood group is AB and Maureen's blood group is O. If they decide to have children, what are the possible blood groups of their offspring? So Maureen is O, and we show O here. This is a reminder. We show O as double recessive. So we're going to put little i's, right? And then here for Michael, we know it's AB, and AB can only be IA and IB. So we're going to cross these and we're going to get A-I, and put it here. B-I, and put it here. A-I here, and B-I here. So what we're gonna have is 50% AI, and that's our genotype. And the phenotype is gonna be blood type A, and that's because A is dominant over I, over O, okay? And then for the other half, we're gonna have 50% blood type B, and that's the phenotype. And our genotype is this, okay? And then for this question, we know Danielle has blood group O, and her father, Jamie, is blood group A. They found, they believe that they found Danielle's long lost mother, and they want to do this Punnett square to find if that's her real mother. And we know that person is blood group A. So for this, we're gonna think that, okay, so we know it's A. So let's say it's homozygous for blood group A. So that woman has big A, or A. And then we know the father has the genotype AI. So we're gonna cross them. We're gonna have A. We're gonna have A. So we see it's 100% A, and Danielle is blood group O. So this does not work, okay? So if you don't want to do this, just remember that if you want if you have an offspring with blood group O, both parents should have one I, little I, and that's because it's a recessive trait. It can only be O if there are two little i's, okay? So here, we're gonna assume both parents are heterozygous. So we're gonna have A little I, and then we're gonna cross them. And we see this is double A, O, and this is O. So we get O here, and we have 25% chance to have blood group O in this situation. So we can say, yes, this woman can be Danielle's mother, but only if she has this genotype. So A, O, okay? I hope this helped. There are no more questions. If you guys need any clarification, please feel free to email any of us. Good luck on your exams.
Hi guys, my name is Katherine and I'm one of you guys' TAs for this class this semester. And today we're going to be talking about evolution and natural selection. So evolution is defined as descent with modification. It is the changes in genetic makeup of a population over time. And a population is an interbreeding group of individuals of the same species that live in the same area. And something important to note is that populations evolve over time and not individuals. Genotype versus phenotype. So a genotype is a set of genes that an organism carries. So a good way to remember this is that genotype and genes both start with the letters GEN, and phenotype is the observable expression of those genes, which are influenced by both genotype and the environment. So both phenotype and physical traits start with the letters PH. So that's also another way to remember that. And then example to show genotype and phenotype is with two alleles, big B and little b. And let's say that big B is dominant for blue eyes and little b is recessive for green eyes. So the genotypes possible here are big B, big B, big B, little b, and little b, little b. So there are 3 genotypes possible. But with these 3 genotypes, there are only 2 phenotypes possible, and these would be blue eyes and green eyes. There are different mechanisms of evolution, and these involve natural selection, sexual selection, mutations, genetic drift, migration, and artificial selection. So there are different requirements for natural selection. The first one is that there is genetic variation for the trait. And so if there's no genetic variation, the population would remain stagnant with its traits, and no traits would be favored over the others. They would all be the same. So this wouldn't be natural selection. The second requirement is that the trait is heritable, meaning it can be passed from parent to offspring. And this is also not great because let's say there's an organism who has super strength, but it is infertile. And so it's not able to pass down this super strength since it's infertile. So that wouldn't be applicable to natural selection. The third one is that there are differences in reproductive success based on traits. So without selective pressures, all traits would be favored equally and natural selection would not be happening. So natural selection operates on the phenotypes or the traits of individuals. And this can be a confusing concept because natural selection, although it does operate on individuals, evolution operates on populations. So the evolutionary consequences of natural selection affects the population, but natural selection itself operates on the individuals. So selecting forces are predators, limited resources such as food, habitat, or mates, and this leads to competition, so there may be more individuals and resources available. And something important to remember is that individuals do not choose to evolve or undergo natural selection. Sexual selection. Sexual selection entails the selection of traits that influence access to mating. So traits that increase opportunities to reproduce, sometimes at cost to survival, are favored. Does this trait make one more attractive as a mating prospect to the opposite sex? Does this trait allow one to gain access to mates? So in the example below with the image, this male has very striking coloration, but this coloration also makes it more vulnerable to predators. But it also attracts more mates. And so this trait is still beneficial as long as the males with the trait produce more offspring than those who lack the trait. So there are different types of sexual selection. So there's intersexual selection, and this occurs as a result of interactions between males and females of a species. One sex displays traits or behaviors to attract the other sex. So the previous example was an example of intersexual selection, because that male bird was showing bright colors. And other examples of this include mating calls or courtship dances. And in the image below, you can see a peacock with all of its feathers. And this is also a way that they use to attract mates. And another type of sexual selection is intrasexual selection. And this occurs as a result of interactions between members of the same sex. So competition is common between males. So they fight for dominance, and this dominance allows them to have the female. So in the image below, you can see different examples of these males fighting. Mutations are alterations in the DNA sequence. They can have different effects on the individual depending on the type of mutation and the gene affected. Some are good, some are bad, but most are neutral. Mutations can be inherited or acquired throughout an individual's lifetime, and some can be spread to offspring. Genetic drift. Genetic drift is a change in a population's allele frequencies based on chance. So there are 2 different types, and the first one is founder effect, and this is when a few individuals from the population start a new population with different allele frequencies than the original population. So The bottom left image showcases this. And as you can see, there is a parent population and a small group of those from the parent population move away and they create their own population. And once this grows and develops, the new population has a different allele frequency than the original population. Next is bottleneck event. And bottleneck event is when a population is severely reduced due to a catastrophic event. And this can be something like a tsunami, a fire, an earthquake. So there's a lot of different things that can happen there to cause this bottleneck, and that is shown in the bottom right image. So migration is the movement of genes from one population or geographic region to another. And in this example, there are two populations split or separated by a mountain. On the right are the brown beetles, and on the left are the white beetles. And they have different genotypes. And one day, a brown beetle goes over the mountain and into the population of the white beetles. And so this introduces new genetic material into that population. Artificial selection is the selective breeding of organisms by humans for desirable traits. These are often not good for the organism. For example, the chicken is much bigger nowadays than it used to be. And this is because humans have selectively bred for the chicken to be this size as it produces more meat for the poultry industry. However, this creates detrimental effects for the health of the chicken. And another example of this is with pugs. Pugs have also been artificially bred, and this has resulted in severe breathing issues with them since their nose is so smushed. Adaptations and fitness. Adaptation is both a trait and a process. As a trait, it is a feature that is important for an organism's survival and reproduction. As an evolutionary process, it is when a population becomes better suited to its environment. Fitness is a measure of how successful an organism is at passing its genes on to the next generation. So the organism must survive long enough to reach sexual maturity and then reproduce. In the first example, an organism that lives a short life but has several offspring is more fit than an organism that lives a long life with few offspring. So this kind of shows that it really is reproductive success that counts with fitness, not how long an organism lives. What's important is that the organism lives long enough to reproduce, but after that point, how long they live doesn't really matter as long as they have more children. So in the second example, and this was already mentioned before, was that there's an organism that is very strong and able to survive a long time, but it is infertile. This organism has very low success or very low fitness. And this is because it has low reproductive success. And so just really trying to emphasize that reproductive success is very important with fitness. So organisms with the most successful traits or phenotypes tend to have the most offspring. Favorable traits in the population increase over generations. So there are different patterns of natural selection, and these include directional, disruptive, stabilizing, and balancing selection. So directional selection is when selection moves the average phenotype in one direction or the other, and it typically favors an extreme phenotype, and it reduces genetic variation. So it could be a movement towards a particular trait as well. It doesn't have to be a continuous trait like size or trait of coloration. So it could just be whether the organism has the trait or not. And then on the right, there is the lizard example. So here's a lizard, and the ones with long tails look like snakes and scare predators. The longer the tail, the more it looks like a snake. And so that trait is selected for, and this creates directional selection so that the longer traits are selected for. Disruptive selection is when variants at the extreme values are selected for. The middling variants are less favorable, becoming less frequent, and this increases genetic variation. So the example on the right, short tails help keep predators from catching you on the ground. Long tails are good for balance in the trees. Medium tails don't help. So in this example, both of the extremes are beneficial traits versus the moderate trait isn't beneficial. And so this creates the disruptive selection pattern. Stabilizing selection is when selection removes extreme variance from the population. Selection favors the middling variance and reduces phenotypic and genotypic variation. So the example on the right, short tails mess up the cat's balance, long tails drag on the ground, medium tails are the best. So here there's selection against both extremes, and the one in the middle is the best, and so that's the one that is selected for. And this is also seen with infant birth weight. So infant birth weight, it's not good if it's too low or if it's too high. And so there's a stabilizing selection there, as it is best when it is in the middle. And that's also when infant mortality is at its lowest. So that's the one that is selected for. Balancing selection is when no single phenotype is favored in all populations of the species at all times. And this maintains genetic variation. So this is different than stabilizing selection, since stabilizing selection is more extreme and it doesn't include as much variation, versus balancing selection, although it also favors the more moderate phenotype, it is more broad and maintains that genetic variation. So there's some common misconceptions about evolution. The first one is that natural selection occurs to benefit individuals. And the second one is that evolution always occurs for the benefit of the population. These are not true. Sometimes they just kind of happened. Natural selection and evolution are not goal-oriented. They don't occur to benefit individuals or the population. They just happen. So there is no goal behind it. And then the next one is that an adaptation is the result of individuals trying to adjust to their environments. And again, individuals cannot choose the adaptations that they have. It's not something that they can bring on to themselves. The next one is that adaptations arise because they are needed in particular environments. Once again, there are no goals with adaptations in natural selection. So adaptations don't arise because they are needed, because this means that they are goal-driven, and this does not happen. The next one is that individuals can evolve. This is false. Populations can evolve. And then lastly, all mutations contribute to evolution. This isn't true. Some mutations aren't heritable and therefore don't contribute to evolution, but the mutations that are heritable do contribute to evolution. So it just depends on the heritability of the mutation. And another misconception is that evolution is very complicated and a very complex process, but this isn't necessarily true. It can also be more simple. For instance, if there is a population of parrots and there are red and blue parrots, and one day a meteor comes and kills off all of the red parrots, all that's left are the blue parrots, and this is what the new population is. So this is an example of evolution, and so it's really not as complicated as it may seem. Yeah, so I hope this helps, and good luck studying and good luck on the exam. I hope you guys all do well.
Let's go over some evolution sample questions. Katie did a great job with these, and I added some onto hers. You can probably tell which are mine because they're a lot stupider. Just before the Industrial Revolution, there was a population of peppered moths that was lightly colored and camouflaged with a lighter environment they resided in. Once the Industrial Revolution began, the environment started to darken with soot. Locals noticed that light-colored moths were now rarities, and that most moths were dark in area. With knowledge of natural selection, which pattern likely occurred here? So when we're talking about patterns of natural selection, we're looking at a shift in the frequency of a given trait or of a type of trait within a population. So in this case, it looks like we're moving towards one trait or like one extreme, and this would just be directional selection. So in thoroughbred horses, the typical birth weight of a newborn foal is 10% of their mother's weight. If the foal is less than this 10%, they are born premature and weaker. And if they are greater than 10%, it can cause complications for both the foal and the mother during birth. What type of selection pattern is likely being observed with a foal weight? Okay, so let's think about which characteristics in terms of weight are favorable and which are unfavorable. So if it's too low, that's unfavorable, and the fowl likely has a lower fitness because it seems to be weaker, so it'll die at a higher. It'll have a higher mortality rate, and a higher mortality rate will be observed for the fowl when it's greater than 10%. And the mother dying is also very bad for the fowl because then it can't support itself. So what's happening is that the extreme values, right, the low and the high, are being selected against, and what is the most favorable one is the middle one. So we call this one stabilizing selection. So let's talk about the difference between stabilizing selection and balancing selection. So balancing selection is where no particular phenotype within the environment is favored. So you'll see more of less of like a peak shift, and it'll be more evenly spread out. Where both stabilizing, directional, and disruptive selection, you'll see that that there is a peak, a more obvious peak to be observed. Based on the graph shown above, which selection pattern is being shown? So it looks like the dotted line was actually dotted line was the original population, and then the blue one, the blue shaded one, is the population after selection. So we see that the middling variables are not being selected against. So okay, so remember whenever you're looking at a graph, the y is like the amount of individuals present, or the amount of individuals, and then the x-axis would be the trait. So looks like this is on a continuous level. So a continuous trait is like height, is like skin shade, weight, that kind of thing. It's not really distinct traits like eye color, for example. So but whatever. It looks like the extremes are being favored. So that's definitely not stabilizing selection. That's not really directional selection because it's not moving towards one in particular. It's disruptive selection. It disrupts the mean. So yeah. So disruptive selection favors the extremes, so the low and the high. Jacob has the allele for like really long pinky fingers, which might explain why his pinky fingers are disappointingly normal. You know, I went to the zoo the other day, and I was like kind of excited because I haven't been to a zoo in like years. But then once I got to the giraffes, they were like disappointingly short. I feel like, I mean, come on, you know, like they're known for being tall and they're short. Like it's like, okay, let's say there were a bunch of aliens, and then they made like a human zoo, and then you show up and all the humans are like normal looking. I would be really annoyed, you know. Anyways, so what might explain it? Interactions at the gene level. He didn't stretch them enough. The phenotype isn't strong enough to overpower the genotype. Interactions with the environment, lower nutrition, and it looks like E is A, D, and B, maybe. So interactions at the gene level. Okay, so if you remember what we talked about gene products, you have two alleles, and then they interact and they're transcribed or translated maybe to produce some gene product, or maybe not any gene product at all. And all these different interactions determines whether or not they'll actually be expressed. So you might have a dominant one that will be expressed, or you might have a recessive allele which is not expressed. And there are other stuff on the gene level like epigenetics. So that is above genetics. So you might have something covering up a gene, like not affecting the gene itself, but it just can't be transcribed. So that's an interaction at a gene level. So we know that one's true. He didn't stretch them enough. We'll come back to that one. The phenotype isn't strong enough to overpower the genotype. That isn't really how it works. So usually the genotype is expressed, and then that determines the phenotype. So interactions with the environment, like lower nutrition, I would say yes for that one. So over here we're talking about a gene level interaction. This is a gene environment interaction. So based on how you live your life and like the amount of stress you experience, your genes may or may not be expressed. So you may have an allele that makes you very susceptible to getting type 2 diabetes, but by living a healthy lifestyle, so the environment would be eating healthy, whatnot, you might not actually get the phenotype of type 2 diabetes. So if he had lower nutrition, maybe that could have inhibited the growth of his long pinky fingers. And then he didn't stretch them enough. Well, I originally wrote that to be like a stupid answer, but I don't know. Like, it is kind of like an environmental thing. So maybe if he stretched them more, he could have that could have activated the gene, or activated the expression of the gene, and then he could have had the long fingers. So it looks like it would be days. True or false? Phenotypes must be able to be seen. So that is false. So when they teach it when they're younger, they think about, oh, like phenotypes are the observable traits, you know, like your eye color, your hair, right? But it's just really easy to explain physical stuff. But it's not really exactly that. Like, you know, there's behavior, like how reactive an organism is, how aggressive they are, their willingness to have teamwork. So that isn't like a physical trait, it's more of a behavioral trait, but that's still a phenotype. And you might have you know, like the proteins in your body, you can't see those with the naked eyes, right? But they have some effect, so that is a phenotype. So a phenotype is just some characteristic about an organism. So that would be false. Okay, apes with big eyeballs mate a lot more. Damn you, anime. What might the current population look like in 45 years? Okay, so this is the current one. Looks to be about pretty stable population, pretty normal, right? But looks like so eye size. If small eye size is over here, large eye size is over here, we would expect a shift to go over here because individuals are mating. With large eyes are mating more, so then the frequency of their progeny, those with large eyes, will increase, and then they'll continue to be favored, whatnot. So we'd probably look at probably C, a very strident C one. So this would be directional selection. What is the best way to describe evolution? Survival of the fittest, natural selection, descent with modification, Darwinism. Evolution is a process by which individuals change over a long duration of time, at least 40 years. So it's this one, descent with modification. So evolution is simply just a change within a population over time, and these traits are heritable and whatnot. Okay. So survival of the fittest, or usually just used to describe natural selection, is a very popular mechanism of evolution, you know, but it's not the only one. You know, there could be okay, let's say that you had a population and you had a bunch of gingers, and then all the gingers within that area went to a convention for sunblock, and then a meteor hit that place, and then all the gingers died. Well, now the population does not have that the particular alleles that they carried, so the frequency of non-ginger alleles will increase drastically within the population in ratios, and that population definitely did not evolve by natural selection. It just kind of happened, but it's still evolution. Which of the following organisms displays the highest fitness from a biological perspective? So fitness from a biological perspective, that is an organism's a measure of an organism's ability to produce viable offspring. So by viable offspring, that means that offspring that can also reproduce. So let's say that for some reason you have someone, and every time they produce a child, that child is completely infertile. That would mean that the person who produced these children, their alleles would ultimately not be passed on. So that is a low fitness. So let's look at over here. A high fitness would just be within an organism's lifetime, how many viable offspring can they produce? So an impressive male who has mated over 2,790 times, and make sure to use protection. Okay, so this male has mated a lot, but has likely not produced a lot of offspring, you know? Maybe like, I mean, it depends on the method. Okay, we don't need to talk about this. Anyway, a blue whale, which has a calf that survives to reproduce as well once every 2 years. Okay, we'll keep that in mind. Definitely a higher fitness than this one. A sperm donor that contributes to Banks' most popular product. It was purchased 14,000 times, and half of the children produced from it go on to produce children themselves. Okay, well that's high, right? So if half of the children are having, half of the sperm donors' biological children are reproducing themselves, that means that there's 7,000 for one. That's a lot. Okay, a really big spider which lays an egg sac. immediately dies right after. The egg sac hatches 6,000 spiders, all of which reproduce. Okay? Well, 6,000 is a lot, right? But within that spider's lifetime, it only did 6,000. Within the sperm donor's lifetime, he produced 7,000. So at the moment, 3 is the winner. And then Superman with vasectomy, the doing of his arch enemy Lex Luthor. Okay, well, Superman is extremely strong, you know, like he'll survive like anything, you know? But it looks like he can't really produce children. So, very low fitness from a biological perspective, but very fit from a dictionary.com perspective. Yeah, whatever. So, it would be this guy. True or false? In natural selection, organisms actively form adaptations to their environment. That's not true. So organisms don't particularly do that. It happens in a population over time. So while it's true that an adaptation is a trait that is beneficial to a given environment, that is formed between generations. So like, you know, that's natural selection, right? So a selective pressure comes about, you know, and then there are some individuals that have an advantage or have some trait within them that give them an advantage with those selective pressures. So then they're able to have a higher fitness in that environment. They're able to mate more, and eventually the population, there's a higher frequency of the alleles that benefit in that environment. So that adaptation. So adaptation is not really an active process. It just happens in the population over time. I did not explain that well, but I'm tired. False. Okay, true or false, all mutations are beneficial. That's very false. So most mutations are neutral, meaning they don't really affect the organism, and a lot of them are bad. And then a couple of them are good, you know, so a couple of them could serve as adaptations, eventually. Select all that apply. What must be present for evolution by natural selection to occur? Mutation. Not necessarily. So, well, let's talk about variation of phenotypes first. So, the organisms must vary from one another, right? Because if they were all the same, there wouldn't be a particular movement towards, like a trait becoming more frequent or less frequent. So, there could already be variation within a population. Mutation doesn't have to apply that, or give that. Mutation would increase the variation, but it's not the only means. So, we have hereditable traits. So that is needed. So even if like think about Superman, right? So Lex Luthor gave him a vasectomy, right? So even though Superman has really strong traits that he could that are really beneficial in the environment to survive, they're not hereditable anymore. So yeah, they need to actually be inherited for for the population to change over time. Emphasis on over time. Selective pressure is such that some phenotypes have greater reproductive success. That's the longest one. Cool. But, oh no. Oh no. Cool. So, let's think about that some more. So, if there weren't selective pressures, like if the environment wasn't, if the environment just was, you know, safe for everyone, that would mean that, while there are variation in phenotypes, like, you know, things look different, things are different, they're essentially the same favorability, you know, like one isn't favored or disfavored over the other one. So natural selection would not really occur. And random mating would actually go against natural selection, or at least the facet of sexual selection. So there's a lot of sexual selection happening within many organisms, and if you just rate it randomly, you just kind of take out that facet of certain sex-related traits being favored or disfavored. Good luck. Email us if you want anything at all.
Justin, are you doing your homework?
Yeah, ma.
Hi, Joseph.
That's trouble, that's trouble! That's trouble!
Tell me. Is. Can I spin around? Do people do that?
Yeah.
But how do you spin in these? You just like awkwardly clomp around. Okay, that's enough. Okay, stop. I think I need to go to the restroom.
Okay, we'll go to the
Moral dilemma. Should I take the car and run?
You get really close, you can take a look at his head. That's his head. Taffy, take your head, some fish.
Some fish were some fish were happy.
Oh, that was close.
About to attempt an epic drop.
just like.
Drop down, Justin. Drop. Don't do it. Justin, what are you doing? No! Justin!